IL-2 is a cytokine that is essential for immune activation as well as for immune tolerance. Because of this key immunomodulatory role, treatment of patients with IL-2 has been seen as an exciting therapeutic strategy, with IL-2 approved for use or in clinical trials for a wide variety of diseases, from cancer to infections to autoimmune diseases. However, its clinical efficacy has been limited by toxicity and off-target effects, a result of its many functions in the immune system. Under homeostatic conditions, IL-2 expression is normally tightly controlled. We show here, using a wide variety of strategies to detect IL-2, that CD4 conventional T cells are the predominant source of IL-2, in line with previous reports, although diverse cell types are capable of IL-2 expression, including regulatory T cells. To interrogate whether the cellular context in which IL-2 is expressed can affect IL-2 function, we generated a mouse model in which we can turn on IL-2 expression in any cellular lineage. This genetic switch drives a low level of IL-2 expression, much less than what is normally produced by activated T cells, but constitutive in a cell type of our choice. We use this model to systematically interrogate IL-2 cellular networks. Expression of IL-2 by conventional CD4 T cells drove preferential expansion of regulatory T cells in addition to CD8s, as is well established. Conversely, IL-2 production by CD8 T cells preferentially fed into autocrine circuits, which allowed CD8s to escape from Treg-mediated suppression. However, we identified there were also differential costs associated with IL-2 production by CD4 and CD8 T cells. The ability of CD4s to produce IL-2 was detrimental to their competitive fitness, whilst the opposite was true for CD8s. Production of IL-2 by natural killer cells also fed into autocrine circuits, with these cells being the major responders to their own IL-2. On the other hand, IL-2 production by dendritic cells preferentially expanded regulatory T cells, whilst largely unaffecting conventional T cell expansion, which may be of particular importance when trying to promote immune tolerance. However, when IL-2 was produced by B cells, unique cellular circuits of IL-2 function became apparent, which were independent of IL-2 dose. We saw expansion of CD8 FOXP3 positive regulatory T cells, likely a result of these cells being enriched in B cell follicles. We also saw expansion of ILC2, which drove severe eosinophilia through production of IL-5. Interestingly, eosinophilia can be a side effect of IL-2 therapy in humans, suggesting that exogenous administration of IL-2 during therapy overrides the tight segregation of IL-2 from B cell zones under homeostatic conditions. These results highlight the complexity of cytokine networks, with the effect of the cytokine being highly dependent on the source as well as the dose. This knowledge of context-dependent functions of IL-2 has the potential to be utilised for targeted delivery of IL-2 to either enhance or quench particular cellular circuits in a specific disease in order to achieve the desired functions of IL-2 whilst avoiding unwanted side effects.